Thank you for the opportunity to be here, and thank you, Rebecca, for that wonderful introduction to my presentation. Let me just see. Okay, I also have, the standard conflict of interest. Of course, as a consultant, I work for a lot of different food companies. I'm a consultant to the Calorie Control Council as an expert advisor, and same as Rebecca, none of those have any influence really on what I say is what I believe, and based on the scientific literature, and of course my many, I won't say how many, years of working in low-calorie sweeteners. So today I want to describe the similarities and differences of low-calorie sweeteners. You've already heard that this is a really diverse group, and I want to give you a little introduction into how they're just different and explain the metabolic fate, how these actually, what happens once we consume them, because I think that this is really important information in terms of sometimes helping to tease out the evaluation of some of the literature out there, and as Rebecca said, really trying to assess, is this even, biologically plausible? Are the doses relevant, and so on. So there are many different low-calorie sweeteners. These are the ones I'm going to focus on today, and really, I think what's important is for you to think about, as I talk, compare this to, as we're talking about vitamins. Vitamins, we say, have in general, and how quickly would you extrapolate a study on vitamin C to the impact of vitamin D? I mean, it's kind of that different, but we often are not so familiar about the differences. So what do they all have in common? It's actually a pretty short list. They provide sweetness with no or fewer calories compared to sugar. And I would say that's about it in terms of how they are all similar. They do this because of their sweetness intensity. These are the high intensity sweeteners. There are other low calorie sweeteners that do not have this high intensity, but the ones I'm talking today are considered the high intensity sweeteners. So you can see that compared to sugar, they have a greater ability to enhance sweet. So what that means is that you can replace 25 grams of sugar with less than a gram of low-calorie sweetener. So, and on average, about 1/10 of a gram replaces 6 teaspoons of sugar. And I think people often don't realize how little we're actually consuming. And I'm going to blame that on these little packets. Because you open up this packet and you can see how much you're putting into your tea, and you kind of think, yeah, that's at least a consumer, is how much of the low-calorie sweetener is in these packets. But if you actually read the ingredient list, over 90% of what is in here is the filler, because otherwise you'd rip open this packet and nothing would come out. You'd rip open another and you just couldn't measure it. So that dilution is for the purpose of making it easier for the consumer to actually measure it. But I think it contributes to the misconception of how much sweetener of these high intensity sweeteners we actually consume. So how do they differ? Well, this is a much longer list, taste profile, stability, manufacturing, source, and so on. The two I'm going to focus on today are the chemical form and mostly on the metabolic fate. And as was mentioned, there's extensive studies on the metabolic fate, starting right at the very beginning of the regulatory approval of these sweeteners. So there's studies done in number of different animal species before we even start, and then of course in the human as well. So this isn't unknown. This is well-known, well-documented information that is available. Unfortunately, it's often not used, as I'm going to illustrate later. Here are the differences. I'm not going to go through this list, but you can just do a quick look through, and you see, we have methylated peptides, we have, diterpenes, we have various different compounds. And why that is important for today's talk is because that affects how it moves through the body. So metabolic fate basically is, can we find out what happens? Once it goes in our mouth, track it. Where is it going? Is it digested by the digestive enzymes in the small intestine? Where and how much is actually absorbed and reaches the bloodstream and therefore the rest of the body? Where all does it go? What's the distribution? Does it cross the placenta? Where, does it reach the brain? Where does it go? And then what's the transformation or the metabolism? Is it simply excreted without any change, or what is that metabolic pathway? And how do we get rid of it? How quickly and where? So this is all from a recent paper in Nutrition Reviews on the metabolic fate, and so all of these figures are from that paper. So in each case, just kind of going through them, aspartame, of course, is that dipeptide that has the methyl groups attached. Let's see if I can get this little, oh yeah, okay, so we have the aspartame as that 2 amino acids and the methyl group attached. Once you eat it, very quickly there is digestion. So it is completely digested into those two amino acids and the methyl group. There is zero absorption of aspartame, so that means there is no aspartame in the blood. That automatically will tell you something in terms of what is the potential for aspartame as a whole molecule to have an effect on the liver, for example. What does enter the liver is methanol, where it is metabolized, and the excretion of methanol is either used in the body or else it is excreted in the air. The amino acids are utilized for protein synthesis, metabolism, and so on, same as those amino acids, whether it came from the Greek yogurt or eggs you had this morning. And the methanol is used the same, whether it's coming from aspartame or whether it's coming from fruits and vegetables, orange juice, tomato juice. All of the fruits and vegetables have that methyl group that becomes methanol when it goes through the digestive tract. So what's important is that you have absorption very efficiently as the breakdown products, and there is no aspartame as a whole going into the large into the colon. Cyclamate. Cyclamate is a very different compound here, and here we have incomplete absorption from the small intestine. That is absorbed simply is not changed, it is excreted unchanged into the urine. The unabsorbed cyclamate that doesn't get absorbed in the small intestine enters the colon. And here we have kind of a unique situation. We talked about individual differences, and cyclamate is one of those compounds where there's clear evidence for human differences. 80% of the population are what we call non-converters. They simply take that cyclamate that is in the colon, they don't metabolize it, but they excrete it unchanged in the feces. In 20% of individuals have colonic microflora that can convert that cyclamate into cyclohexamine. And then that cyclohexamine is absorbed and excreted in the urine. So and even within this 20% of people that can do that metabolism, there's a huge variation within within that 20%, and a huge variation within each individual, because it depends on what else are you eating, are you constipated or not, because that will affect how long it's sitting in the colon. So there's huge variation in this. And all of that information has been extremely well documented, and extensive studies have been done on this variability, depending on diet, depending on age, and this is all being carefully considered in terms of establishing the acceptable daily intake for cyclamate. I've put these two together, acesulfame K and saccharin, because these are two that act very similarly. They're both pretty small compounds. They are pretty effectively absorbed in the small intestine. ACE-K is completely and very rapidly absorbed, actually. So it very quickly goes into the systemic circulation. There is no metabolism. It is simply excreted through the urine. At high doses, some saccharin can enter the large intestine and be acted upon by the colonic microflora. Even though we just had this big paper that came out and reached lots of headlines, actually we've known that for a long time. That was published in the 80s. It was evaluated and again considered. As soon as you have something going into the colon, for us toxicologists, red flags go up. We know that things happen down there. And so there are studies that are conducted. Okay, and so that of the saccharin that does enter the feces, again, there is no change, there's no metabolites, it is simply excreted, and same that is absorbed goes through the urine. Stevioglycosides. Okay, this one's a little bit more complex. Here we have stevioglycosides, as many of you know, are composed of steviol as the backbone, and then there is a whole wide variety of different glycoside linkages that are attached to that steviol backbone to give you all the different stevioglycosides that we have. They all actually, though, go through the same pathway of metabolism. So here you have nothing going on in the small intestine. There's no action by the digestive enzymes. There's zero absorption of the entire of the stevioglycoside. So everything goes into the large intestine. Everything enters the colon. And that's where the action is. Okay, so once it gets down into the large intestine, the colonic microflora chew up those glucose linkages, and what you have left in all cases for all of the stevioglycosides is the absorption of steviol. And that's why we have the ADI that is based on steviol equivalents, because for that whole group, what is entering the body is the steviol, and that is what we are talking about in terms of systemic exposure. That steviol goes to the liver, and there is a very standard metabolism change where it changes to a glucuronide that facilitates the excretion through the kidneys. So here we have, again, quite a different effect. Sucralose. Okay, now this is again zero absorption, or very little, I should say, absorption. The majority of sucralose shoots right through into the colon. We have numerous studies again conducted as soon as it is realized that it's going into the colon. A lot of studies done. Can the colonic microflora utilize sucralose in any way? And to date, there's no evidence that there is any metabolism by the colonic microflora of sucralose. There's no digestion, so that is, of course, why it's low calorie. We don't break it down into the two disaccharides, but it is not digested for any calories. There is a little bit of absorption, about 15% is absorbed of sucralose. Most of it is simply excreted into the urine unchanged. A very small amount, like 2% of this 15%, so calculate what that percent is metabolized and it's also excreted into the urine. But sucralose is one that takes a little bit longer. Its rate of excretion is slower than some of the other ones that are quickly absorbed into the blood and excreted in the urine. So this is kind of a summary, and I'm not going to go through this because I've just, and everybody's taking pictures, thank you very much, but if you'd actually like this one, I'll give you my email at the end, and I'm happy to send any of these slides to you. So, but it's just a snapshot really to show you that there's differences in these, and trying to apply, say that a metabolic study on one tells you something about the other is really rather naive, is a polite way of saying it. So how can we use this information then? I'm going to try to give you a few examples. One is that we can actually know, based on how much is absorbed and where it goes, how much of it actually, what would be our daily systemic exposure? So how much would our liver or our blood actually see on a daily basis? And this is, again, it's in our paper, we describe how we do this estimation, but it's assuming that all of your daily intake of low-calorie sweetener was from one particular sweetener. And often that's not the case. We have certain type of sweeteners that work better in beverages, and some that work better in baking, and some that people like better in their tea. So most often people are using kind of a mixture of different ones. But if we had all of our exposure from a single low-calorie sweetener, how much would we actually, what would be our systemic exposure? And you can see, again, it's small, right? It's less than 100 milligrams in all cases. And I think that why that's important is when you start looking at other studies and what are the amounts, especially animal studies and studies where they're adding it to cells. I mean, does this really make sense in terms of, we know how much the body would normally see. And I think it is often not considered. One of the other important factors is that the metabolism really illustrates that there's no likely site of interaction of low-calorie blends. And this is often a question I get: what do I think about the safety of the blends? Again, this was already mentioned a little bit already by Rebecca, but there's a lot of benefits, and it improves the synergistic enhancement, so the blends often taste better. They often have improved quality of sweetness, so often there's less used with a blend than if you're trying to do the sweetness with just one particular sweetener. And there's a lot of common mixtures. Probably one of the most common one is aspartame and ACE-K. And as I just showed you, they are very different in terms of how they're absorbed, whether they're digested or not, and how they're excreted. Same with sucralose and ACE-K. Sucralose going straight through the colon. ACE-K is completely absorbed. So you have very different mechanisms of absorption, no metabolism. So the possibility of one to affect the other just is not there. And that's actually been, shown in a recent study looking at sucralose and ACE-K to confirm no difference in plasma levels when they were given alone or in combination. Well, I mean, that really was exactly what you would predict if you looked at the two. So yeah, we did the study, but it's like, yeah, okay. I think this is a really interesting commentary that was published by Dr., I'm not sure how to say, Glendinning, on the factors contributing to confusion and controversies about low-calorie sweeteners. And I just sort of picked out a couple here that I think are really relevant to the talk today. Most studies have not considered the divergent metabolic fates of the low-calorie sweeteners, but treat them all as interchangeable. And I agree that they're interchangeable if we're talking about whether or not they're replacing sugar. Yeah, then okay, and whether or not, what they could have, impact they could have in terms of caloric intake and so on. Then saying they're interchangeable is fine. When we're talking about potential metabolic effects or biological effects, then I disagree that they are, that you can say one predicts the effects of the other. They're often, many studies are confounded by biased subject selection, which is of course more of a clinical, but a lack of clinical control groups. And I see that so often, even in many of the published studies trying to assert that there's a biological effect. The kinds of toxicology studies that Rebecca has talked about are based on pre-assessed, or I should say, predetermined controlled studies where the design is established to ensure that you have the correct negative controls, positive controls, to better allow you to interpret the studies. And so often in a lot of these other studies, there's either no control group or an inappropriate control group. And one good example is for the low-calorie sweeteners. If they are giving high doses of a tabletop sweetener and not considering the carrier that is 90% or more of what's in this package, then how can you attribute the effects to only the low-calorie sweetener and not consider that you've added a whole lot of dextrin to that diet? So these results, and these are his words, result in considerable controversy and rancor in the field, and I think that is really true. I have found also two other factors that they don't consider or discuss existing research and data. And most frustrating to me is that speculations and hypotheses suddenly become fact. And I'm going to give you a couple examples. This is the one that we're probably going to hear about later in the talk on the microbiota, where all the studies on the microflora were done with saccharin and suddenly applied to every low-calorie sweetener. Kind of forgot that we knew about that already, that those studies were done in the 80s. Not one of those studies were cited in that paper in 2014. And no consideration about the other low-calorie sweeteners, often don't even get to the colon, or they're completely metabolized and so on. The other one, just most recent, is this title: Intestinal Metabolism and Bioaccumulation of Sucralose. Bioaccumulation of sucralose in the adipose tissue in the rats. They didn't measure intestinal metabolism. Nothing in terms of the intestine was even investigated. What they did was identified two peaks in the extracts of feces, and these are proposed metabolites of sucralose. Did not determine the structure, but guessing what the structure is, and then guessing that this is the result of intestinal metabolism. And yet it's the title of the paper. Bioaccumulation, also not measured. Bioaccumulation means increase over time. They only measured the sucralose in adipose tissue in 4 out of the 10 rats at one time point. That is a snapshot in time. That doesn't tell you that there's any accumulation. It tells you what it was at that point in time, which is, so this is really frustrating. This is now the title, this is the abstract, and this is now going to be cited as sucralose is metabolized by the intestine, microflora, and accumulates in the adipose tissue, when in fact we have zero evidence of that. Little frustration coming out in my voice. You can hear that? Yeah, well, that's kind of how it is. So also consider the concentration. We know what the typical blood levels are following human consumption, and always in the range of nanograms per ml. And so we know that in terms of if that's what's in the blood, that's the tissue is going to see, and potentially that's what the cells in those tissues are going to see. For some reason, this is forgotten, and we see so many of these mechanistic in vitro studies using concentrations, 10 to the third, 10 to the sixth higher. Whatever concentration will get an effect is the concentration you use. And that's just unrealistic. The results are not biologically meaningful. You have to be able to get people to only publish those studies in which we're really using realistic concentrations and are actually identifying any, if there are any, effects that might occur in a human population. And I mean, most of the time, there would be none, which is why we don't have those publications, right? So, in conclusion, low-calorie sweeteners represent a very diverse group of compounds, and their differences in the metabolic fate are really important to understand if in terms of looking at some of this research and being able to determine whether or not it is even biologically plausible, and whether the study design and data interpretation are appropriate. So I look forward to the next rest of the afternoon and discussions, and thank you very much.